So we have the envelope, which is the outer layer of the cell. The cell is going to be HIV. Coming off of the outer layer is first the transmembrane glycoprotein, also known as TM. Coming off of the TM is the surface glycoprotein. Then you have the matrix protein to help maintain viral structure. Then we have the capsid protein, which is the same as the matrix protein. Inside the core are 2 copies of the RNA and 3 enzymes that are reverse transcripts and protease and integrase. This is all part of the HIV cell. The HIV attaches to the host cell called CD4, that is the T helper cell. The T helper cell is a white blood cell that fights infection. In the attachment to the CD4 receptor, there has to be a co-receptor. The co-receptor differs for any host cells. After attached HIV envelope in the CD4 membrane fuse, this causes entry into the cell, which is CD4 cell. Once HIV enters into the cell, RNA is then released into the cytoplasm of the cell. Reverse transcripts makes DNA a copy of the viral RNA genome. When this process is happening, the RNA strand disappears. Then a complementary DNA strand is added by the reverse transcript. The ends of the DNA are joined but are not bonded together. Once they are joined together, the DNA moves inside the nucleus, and then it is inserted into the CD4 nucleus. The intergrand viral DNA is now referred as prival DNA. The DNA can stay within the CD4 cell, or RNA can also be synthesized from DNA. Then you will get the messenger RNA and a viral genome RNA. The messenger RNA is translated with the viral enzymes and structural proteins. Some of the functional proteins are formed by cleavage of a polyprotein by the enzyme protease. The protease inhibitors interfere with this step. The TM are inserted into the cell membrane, and then the structural proteins surrounded the viral RNA to form a new core. It is then released by budding.